Okay, recording is on, and now regarding the course, do you have any questions of the practicalities or the lecture of databases, or maybe the homework? Can we get the ball rolling somehow? Somehow that we get some discussion.
The final deadline of the big homework is going to be this Friday. Is it a hard
Pardon?
deadline?
Can you repeat that, please?
Yeah, sorry. So for the big homework, the deadline is going to be this Friday.
Yes, that is the suggested deadline, so that you keep up with the previous topics before the next ones come. So
Okay.
they've been set like on the Friday after the lecture for each of the homeworks.
Yeah, but it's not a hard deadline. So if it's going
It's
to
not
be
a hard
submitted on
deadline, exactly.
Saturday
So
morning, that's fine. Okay.
basically, you can submit everything in December. The final deadline for the project would be more or less
Okay, thank you.
before Christmas, so maybe December 20th. But the suggested deadline again is November 22nd. Now, if I'm opening a PDF, do you see it in the shared screen?
Yes.
Very good. Yeah. So in the schedule here, the biggest tasks in the whole course are the final project. Okay, November 17th, actually, that's what I've suggested here, but this is open to discussion. And so this is the biggest task. It would be requiring like several days of full-time work, or a couple of days of full-time work. And the other big thing is the multiple sequence alignment portfolio. This is an exercise which I've had in bigger groups as a peer-reviewed workshop project, so that the people are submitting the work and then reading the reports of 2 or 3 other students. But with so few students and with different kind of scheduling pressures for everybody, it wouldn't work to have a strict deadline for submitting the reports. So I'll take the burden of grading your work, but there will be a self-evaluation and pretty good instructions of how much each part in the task is worth. And so if you don't have time to do all the parts or the energy to do all the parts, so it's okay to do something less, but you would be just listing what you did and how well. It's pretty well instructed in there. And then the so-called major homework things, as you may have seen, do not require more than a couple of hours, but still they are essential for your learning. My philosophy is to learn by doing, and for a thing like bioinformatics, where you have lots of online tools, the best way to learn the tools obviously is to use them. But the nice side effect is that while you are doing the analysis, you start to think more profoundly about the theory as well. So it works in many aspects. And so that's why we are quite heavy on the assignments and not so much on the theory. But if you want to learn in a theoretical style, if that fits your way of learning, and if you are curious so that you want to go further, so we do provide the resources. There are plenty of links in the course which are kind of semi-optional, that I'm not requiring you to read fully every web page which is there, but they are for the purpose of finding information. And obviously the help facilities in the services themselves, like Uniprot and NCBI, are pretty large masses of text where you easily find what you need to find for most exercises. Have any of you been doing the first homework yet?
Yes, I already
Anybody?
started.
Okay, that's
Me
very
too.
good. And and let's see. I saw some of the biological databases discussions last week, and oh yes, there are plenty of more now. By people posting out of the 7 you are. So this is really nice. Yeah. So I guess one of the take-home lessons for the first week's class is that there are really many databases. The purpose of these minor exercises was to make you browse and try to see if there is something that interests you personally, and many of you have actually found something that relates to your previous experience or something you might be needing in the future. And one of you mentioned the AlphaFold database, which is an absolutely huge thing, something that has basically solved the famous protein folding problem. We'll talk more about that in our structural bioinformatics course, but that is really a revolution, so that the predictions give us models which are pretty much the same quality as actual crystallographic models or experiment-based models we previously had. And the number of known proteins has exploded 1,000-fold. So coming from about 300,000, we are now at 200 million. So it's really a lot that we have there. And these databases are really of different sizes and shapes. So AlphaFold database is huge in impact and also huge in the content. Think about 200 million coordinate files that are some hundreds of kilobases each. You don't want to download it in your computer, and you probably couldn't do it in one afternoon. So there's a lot, but then you have some very specific databases that might only have a few entries. For instance, my ex-boss in Tampere had an obsession about diseases of innate immunity or well, actually of any kind of immunity. So diseases which led to immune deficiencies. And he had a target called Bruton's tyrosine kinase, which we also see in some of our lectures, at least in the structural bioinformatics. And this is one for which hundreds of mutations are known. So he collected mutation databases that had the full information, whatever was known of the severity of the disease with that mutation, articles where it was described, etc. And I think he had something like more than 100 mutation databases at the time, so that each immunodeficiency related protein had a database of its own, and some of those databases had just one or two mutations, but they were made to the same format as the biggest ones, and they were expected to grow. But then the funding kind of dwindled, and Mr. Vihinen or Dr. Vihinen moved to Sweden and moved to other projects, so they haven't been really updated in the last more than 10 years, maybe almost 15 years now. Well, could be 12 years, but still. So that was an example of very small databases. But nevertheless, for the person who is looking for all possible information in a specific immune deficiency, if the causative gene and protein are known, so it's very nice that there is information of all the known mutations. And for many proteins, there were available structures, and now again with AlphaFold, the database would be hugely improved because every mutation could be displayed in its structural context, and that might help in figuring out the mechanism, and maybe if well, if a protein is broken, it's very hard to find a drug that would make it work. So it wouldn't be helpful for drug design. But anyway, for understanding the disease mechanisms, it's certainly something that made the effort worthwhile.
Can I have a question?
Sure.
It's just yeah.
Actually,
So Mario is here. I I'm not really familiar with the nucleic acid databases. Like I use more often protein related stuff.
mhm.
Yeah, but so when I check the homework and I check there is some early question like we have to have some information about PT and gene or something like this. Yeah, and that's the name of the gene. And
Yeah. Mhm.
I see so many information on the NCBI database related to this, and most of them I'm not sure what is it about. So if it's boring to the others, the explanation can be short. But
Well,
yeah, for me, like
but
many arrows on the I don't know on the line, and then so many graphical stuff, and I'm not sure. Yeah.
yeah, the NCBI website is maybe not the optimal thing, and And I share your sentiment about proteins being the cool thing and the guys who get things done, and DNA is just the blueprint that is carrying the task of having the instructions how to build the proteins, and also having regulatory elements that help to manage the whole thing in a very complicated multicellular organism, having different kind of gene regulation for different tissues, and orchestrating the individual development of cells and tissues, and so there are definitely cool aspects in the DNA, but they are not as clear as proteins, and less is known. But they are important for the purpose of being the primary information for proteins. Very few proteins have been sequenced on protein level, and everything comes from conceptual translation of the DNA, and therefore the DNA databases are important as well. And actually, I was I was selecting one question. We had a brief discussion with Christina. She's not here now, but she was asking about the question of the gene length, and she was discovering two different lengths depending on where she looked. And did any of you have the same observation?
Yes, I was wondering about that actually.
Okay. So yeah, so I think the big lesson there is that there is so much of bioinformatics data that has been assembled at different times, and by different institutions, and maybe by different standards, so that you will find conflicting information, and sometimes you just need to do things like align the DNA sequences yourself to the genome. Ensemble genome browser is perfect for that, and it's way better than the NCBI genome browser in the user interface. So this was like just a quick look at what kinds of data there is for DNA, and what I like in NCBI is that you from the gene page they have pretty nice links to the RefSeq elements, which would be the RefSeq messenger RNAs and the RefSeq proteins. So it leads to all the known information for all the biological molecules related to that gene. But to return to the reason for the different length, so I have the impression that some shorter definitions of the gene start from the first triplet of translation, so the initiating methionine, the ATG codon, and then they go on and across all the exons coding for the protein and introns which are falling in between, and then end the count at the stop codon in the last exon. But then the mRNAs are longer than just the coding sequence. There are untranslated 5' regions before the start of translation and untranslated 3' region in the other end. So the longer lengths for a gene would be taking the position of the exon starting from the start of translation and ending at the polyadenylation site where the poly A is added in the messenger RNA when it maturing. I'm not sure how many of you have done biology at a sufficient level so that you are making sense what I'm telling you. Is this going to too much detail, or is this just okay based on your school biology or whatever courses you've taken at the university level? If you feel that you are not quite okay, I have a quiz which I've been using in other courses to test your level of molecular biology skills in various aspects. So I could set it up if there is anybody wishing for it, and so you can take it and see what area gives you most wrong answers and maybe target your learning based on that. Nobody's saying anything. That's a difficult topic. At least any Finn would be very hesitant to say in a public place with unknown people that there's something I don't know or something I can't do.
Actually, I'd like to check that quiz.
Okay. Yeah.
I would appreciate that. Thank you.
Okay. Yeah.
Thank you. Thank
Very
you.
brave. Thank you so much. So I'll set it up this week. And by the way, thinking of overcommitting, so I promised to make a re-recording of the course introduction, but I think we are pretty well set up so that you know what you're expected to do, and the syllabus is giving you the full details of this year's dates and this year's topics. What is new is that we have the summary of tasks, and tomorrow morning as the first thing I should be making a similar summary for week 2, but anyway, what it contains is pretty much the same things that you should be looking at, watching the 2 videos here, and then there is a summary of blast details, which is going to a little bit more detail than what we show here, but because it's kind of overlapping, so I decided not to have the video on display. Likewise, this long video of blast searching is just like the same material but twice that long. So this is a nice concise video we made for a course in Ireland. Likewise, the video for databases here, which is here, it is part of another course. So we are trying to reuse material when we have good videos for anything. So the blast searching is a little bit more extensive in terms of materials, but then in terms of the content, it is a much narrower thing, just one search tool. But you need to go a little bit deeper into it so that you will become informed users But we'll talk more about that when you have done your learning here. Any other comments?
Hello. Sorry, I just have some questions because I already started the homework and I watched the lecture, and so there are
Yeah.
just things that I want to clarify. For example, one of them is like when you in databases, for example, when you say that it's automatically annotated, is it the same? Does it mean the same as predicted? Like when you see in databases, do they mean the same thing?
Well, computational annotation is sometimes a prediction, sometimes it is an extension of an experimentally derived knowledge into other proteins which are assumed to work the same. So you could say they are predictions as well. But it's obvious that there is no way that the scientists on Earth could do experiments on all the 200 million proteins we have in the UniProt database. But we do have model organisms which have been investigated heavily, like human and mouse and zebrafish, fruit fly for genetics, E. coli for bacteria, Saccharomyces cerevisiae, the baker's yeast for a single-celled eukaryote, the yeast. So when we have experimental knowledge of proteins in these organisms, so we can easily identify computationally homologs of all proteins, so proteins which are very likely to be derived from a common ancestor. And in the homologs, we can then outline orthologs, which are like the same gene in different species, and then we have paralogs, which are different genes but with a similar structure and a common origin, and most of the time similar functions as well. So transferring the functions on them is a way to do way to go to save money instead of doing the same experiments for the rat homolog, the gorilla homolog, the chimpanzee homolog, the dog homolog, you name it. So we can assume that the metal binding site in a carbonic anhydrase enzyme, when we identify it by sequence comparison, by sequence alignment, we can assume that the aligned histidine residues in similar positions in all of those proteins, they all are responsible for binding zinc. So it is kind of transfer of knowledge to similar objects, but this might be getting risky at some point when you are just transferring this information to objects that are further and further away from the original. Maybe sometimes we have an enzyme which looks like an alpha carbonic anhydrase, but it's actually different in its catalyzed reaction. We know that many CAs have side reactions that they can do other things than the main reaction, which is combining carbon dioxide with water or breaking down bicarbonate ions to create carbon dioxide. So there are many other reactions that are happening with this enzyme. Maybe we have an enzyme which only does the kind of side job. So it's been specialized, and where that happens in the spectrum of sequence similarity is a big question. But that is why experiments are being carried out in different kingdoms of life, so that we don't try to do conclusions like based on mouse experiments, humans are small furry creatures that like cheese. So there's a limit on how far you can generalize your observations, and that happens with molecules as well as on organism level. But then we have many kinds of computational tools for finding features in proteins. So, for instance, the transmembrane helix annotations. I really would love to share my screen. Let's see if I can change into the full screen now. Hmm. No, my share button is disabled if I'm trying to do the full screen. Okay, found it. Yes, I'm sharing my screen. Now you're supposed to see my full screen here. So I happen to have some sequence alignments. Can you see something like a colored carpet with lots of stripes of different colors?
Yes, we can.
This is a multiple sequence alignment of which you will be hearing a lot, but just to make my point of the transfer of functions, we know that in this motif of histidine, something histidine, this is one letter code for amino acid. So these two are zinc binding amino acids, and then there's a third one which is further down here. It is in this glutamic acid, a hydrophobic residue plus H. So this is the environment where we have the third one. So if we follow here this HXH motif, so you can see that the X is changing. We have a different amino acid which is coming between the histidines, but the histidines are there. And likewise in the third histidine, so you can see that the histidine is there, and well, this happens to be a fully conserved EVH motif, but it could work just as well even if this valine instead might be a leucine or a methionine or an isoleucine. All of these are medium-sized hydrophobic amino acids. So either of them, any of them would work in this environment. Then you can see things like a C here, a cysteine, which you may remember is important for making disulfides, and especially conserved Cs are very likely to be involved in disulfides. And if we look at an AlphaFold model, we might then find which C it is binding to. And there's one fully conserved C. And looking at the models, seeing which Cs are pairing with what, and finding if those Cs are conserved in other proteins of the family, we can infer that we have disulfide. So here, looking here at up, we can see that this is column 233 in the alignment, and it's amino acid 135 in the protein, which I just selected. And in this protein, it is amino acid 169, and we would annotate it to a different number, and so on. But this is one way of transferring information. Did any of you look at the protein data bank in the databases? The homework is more strongly geared towards NCBI and UniProt, which are the major sequence databases. And actually, NCBI is a big site that is a home to multiple sequence databases. So they have the full GenBank and then the smaller subset RefSeq, which is like a selected collection of reference sequences. So, but PDB, it's different in that it's a collection of three-dimensional structures of proteins derived from experiments. Nobody's confessing. Well, if I remember correctly, so the homework did have a little bit on the on the structures as well. Actually, okay, yes, there are 2 questions for protein database PDB. But did you notice the AlphaFold models in UniProt? Does it ring a bell? No. Oh, come on. Type UniProt. UniProt. So let's see what we could have. A 6 zebrafish. So, but by the way, one thing that is hard to see if you are just scrolling up and scrolling down is that you are losing the, well, okay, now we do have these tabs up here, but in an earlier version, once you started scrolling, you didn't realize that you are only in the entry tab, and some of the things are here in the feature tab. So features are annotated positional items. So, for instance, here we have a signal peptide. If you're aware of some proteins that move between different targets inside the cell and proteins that are being secreted out of the cell or being sent to the outer surface of the cell, most of these targets are sent to the endoplasmic reticulum and they have the signal peptide. And the signal peptides have specific features so that let's see, if we click on this, do we get the sequence? No, we just get the position, but it tells us that this has been based on automatic annotation. And let's see if we can follow this and find what are the rules there. I can't find what is the method they used, but for some proteins you can, okay, now this is using a prosite signature. So based on kind of protein patterns for signal peptides, they can recognize these. But for bacterial proteins, they use often a sequence-based prediction method, which is finding the signal peptide and the cutting site for that. And also publications, if you are looking for the articles, so you would find them here in a separate list, and yours truly is actually getting his 5 seconds of fame by showing an article being in UniProt database. But you have these things here in separate tabs, but the entry tab is where we have most of the biologically important and practically useful information. And the sidebar here is helping you to jump to the sequence. And there was a question of getting a fast A sequence, and there used to be there used to be a link called FASTA, but for a non-specialist, they don't really get what it means. Excuse me. So instead, the same thing happens now through the link called download. And because it's in the sequence query, you can assume that it's downloading the sequence. And it's opening a new tab in the no, it's just a link directly to the download. So this is a FASTA formatted sequence in which we have this greater than sign to indicate that this is a line which shows documentation and is not part of the sequence. The sequence name is anything that comes before the first space, and what comes after is like free format documentation or free format notes. But because this is coming from a database with a regular structure, it is giving specific information. So we have the protein description string here, and then we have the organism, and the OX, I believe, is the organism code in the NCBI taxonomy database. We have gene name, GN, protein existence, the level of information, what we know, whether it's known at the protein level or at the transcript level or only predicted. And the SV, I forget what that was. Well, anyway, so you can get the sequence in an easily usable form. So if you just copy paste from here, you would have a hard time. Well, actually, no. It seems that the database is clever so that the numbers are overlaid on the sequence in a different layer, so that when I'm doing a copy here, so it is only pasting the sequence as a single string. But still, there are no line breaks there, so to make it human readable at the same time, so this FASTA with the lines broken down to 60 characters per line, 60 amino acids per line, that's more convenient for editors and such. And because the software that handles sequences and the web servers which handle sequences, they know about the FASTA format, so they will take anything that is starting after starting at the next line after this heading, and then it's going on all the way until the next greater than symbol. We might have a file which has multiple sequences in FASTA format. That is legal as well. And then it takes whatever is here and filters out all the line feeds, line breaks, and spaces, and tabs, and numbers, and non-sequence characters to get the actual sequence. But I'm kind of digressing now further than we should, but maybe that's helpful to make you understand how you work with biological sequences. And the FASTA format is kind of important, and that is why there is a question about that even in the very first homework. If you need to do a BLAST search, the first thing to do is to find a sequence of your interest, take it in FASTA format, and put it in the text box where your query sequence is going to be the sequence to which you're seeking similarities. But if we go back to, let me come back to see if there are any questions here. No, nobody's been putting anything to the chat. But in the UniProt homepage, you have search tools which help you do some advanced searches. So this is kind of nice in that it has the common things here, but then they have a huge list of fields which you can which you oh sorry they have a huge list of options for doing things. So we have here the basic things first, but then all of the items here in the list of options for searching, if they have the arrowhead, it means that you can expand it. So subcellular localization or subcellular location, you can add a condition for finding transmembrane proteins which have an identified transmembrane helix. Or if you are interested in diseases, so this is kind of confusing. Pathology and biotech, you wouldn't think that this is the same thing as diseases, but anyway, thinking of pathology, it's about diseases and aberrations in the organisms. Okay, so here you can do a disease search, and then it gives you a further option of only accepting experimental information about a protein being involved in some disease. There might be some, or there will be proteins where we have an inferred function, and again, this is where we are getting tricky. So let's say we have anxiety and some proteins which are related to how easily a person is experiencing anxiety, and or drug targets which help in treating anxiety. So how far can we go in the animal kingdom to think about anxiety? Well, I can easily see from my dog that it might be anxious and experience fear and all kinds of things that are similar to human psychology. But what about let's say spiders or flies or maybe inchworms, not to speak of yeast or bacteria. But still there are some plant proteins that are named with something that includes anxiety because they are homologs of an anxiety related protein in human. But you couldn't really think that the plant would be getting anxiety by a mechanism related to that protein, or that the plant becomes less anxious if it's treated with the same drug as the humans. So there's some limit of where you can transfer the information. But if you want to stick to the experimental information and not the inferred information, so you can choose experimental evidence. You might be wondering, where do you get to know gene names? That's another thing. You probably would go for a Google search and try to see what they tell you about official gene names. Let's say human, and they will point you out to the Hugo Gene Nomenclature Committee. This is where you find the authority of deciding on gene names. And once you do a search here, so then you would have a gene name for doing a search at UniProt. But that's something that is good to know, and that holds true for any database. If you want to be targeting specifically, so the advanced search is your help. So maybe if we do a search for anhydrase in human. Well, we are getting a huge amount of hits. We could help this with the trick of adding quotes so that it's only finding the whole string, but it is still bad. So having human here as a keyword doesn't work as well as going for the actual organism field to target the search. So we can use these sidebar filters to do the same effect as going to the advanced search and making an organism based additional search for Homo sapiens. So this gives us about 89 sequences, and if we want to find the good quality CAs, you are probably aware that UniProt has the reviewed and the unreviewed parts, and so maybe carbonic anhydrase, being such a long known enzyme, would be more in the reviewed part. So we are down to 26, but still we have 26 and not just the 15 we should have here. And we can see that we have all kinds of different things. So why is this receptor type tyrosine protein phosphatase there? So let's do a control F for carbonic anhydrase. So there is a domain which is alpha carbonic anhydrase like, but I can tell you that this is not enzymatically active. So this is kind of a false positive. It belongs to the family in a way that it's homologous, but it's lost its function at some point, and it's working as a receptor to some neural molecules. And for many of these proteins which are not carbonic anhydrases themselves, we may have something that is reported, maybe an interaction with carbonic anhydrase. So let's see, binds to carbonic anhydrase like domain of this PTP receptor. Okay, we find the target immediately. So how could we improve this search so that we are only getting the actual carbonic anhydrases? Any suggestions? No, we could obviously, with just 26, we could click through all of them and make the human expert decision and find the ones which are the good ones. But if you had 200, it's getting tedious, and 2000, it's going impossible. So how could you improve this? Maybe we could find the proteins where carbonic anhydrase is in the protein name. So if the main title of the protein is something else, so this tells what is the function of the protein, the identity of the protein. And having the hits here means that carbonic anhydrase was discovered somewhere in the entry text. As you've seen by browsing, these are pretty big essays of biological functions, so you will find all kinds of things there. So going for the going for the protein description. So here is the advanced search. So we can delete the human here and go for adding fields. So going for organism and adding another field which would be going for the protein name. And this is a new search, so we don't have the filter for reviewed Swissprot entries, but now we are down to 16, and you can see that they are all of actual carbonic anhydrases. There are still members in the family which are called carbonic anhydrase related proteins, which are not enzymatically active. So CA8, 10, and 11 are CA related proteins. And we were supposed to find 15 because there are CAs from 1 to 14. So how come it's 15? It's because there is a glitch in the numbering. The CA5 has like proteins 5A and 5B. This is a mitochondrial protein, and instead of having one which was discovered like several years afterwards, instead of having this named CA9 or CA10, whatever was current at the time, they decided to put it into the numbering so that the two mitochondrial CAs are 5A and 5B. Oh well, but then they didn't want to do the same with the cytoplasmic ones. 1, 2, and 3 are cytoplasmic, and then when 7 was discovered to be cytoplasmic, they didn't want to call it 3A or anything. And likewise, 13 is cytoplasmic. But anyway, we have those 1 to 14 adding up to 15 proteins, but we have 16 hits. And now the odd one out is here, and looking at the title, so putative inactive carbonic anhydrase 5B like protein, I think you can see that this is probably not a thing you want. And looking at the lengths, most of these are more than 250. 260 is typical, and then there are some that have additional signal peptides, transmembrane domains, etc., to bring them to more than 300. But this one is more like a pseudogene, which is probably not a true protein. So protein existence uncertain. So you can easily see that this is not it. Not it. Let's see if we can include that in the search. So let's add a field and let's see how the protein existence evidence at protein level. So let's say that we want to have something in other species that would be homologous as the lowest level of evidence. So putting it here and sorry, this is only finding proteins with that evidence. So again, I wonder if we can. No, it only accepts one at a time. Well, let's be happy with this 16 and with the fact that we have one which is derived from a pseudogene. It is a translation of a pseudogene fragment, which is only coding for 2/3 of the full length of CA5B. And there we have the best versions, the reviewed Swissprot versions of every carbonic anhydrase isoform. This is kind of unusual that the reaction is so important that we have 15 enzymes that do the same reaction, or actually 12 enzymes that do the same reaction, and then 3 of these CA-related proteins that have specialized into other non-enzymatic functions. But that was like a quick demo of the work here. And let's look at one more thing, the customize columns, so we could have more columns here on the screen. So let's see if we want to have structures. So let's select 3D and let's see what happens. I'm just looking if there is a save button. No. Let's refresh the screen. Where do we have the 3D column? Oh, come on. Oh, there. It was behind my pop-up screen here. So save, and there we have. Now we have the information of how many 3D structures we have for each of these. So CA6, 1 X-ray based model, 13, 16 models, etc. For CA2, we have 1,000 X-ray based structures. This has been like a sandbox for the crystallographers to create crystals of different mutants, different complexes with drugs and inhibitors. And there are even some neutron diffraction and other methods that have been used. And there are some for which we don't have any models, like the CA-related proteins 10 and 11. And now if we go to CA10 and go to structures, well, there is a structure, but the trick is that the 3D item was only for experimental structures. So this is a model coming from AlphaFold, as you can see from the bottom of the screen. So here there's a link to the AlphaFold database itself to retrieve the coordinates, but here we have a thing which zooms in and out with the wheel of a wheel mouse, and with the right button I can translate it, and with the left button, the primary button, I can translate it, and with the secondary button translates and primary button rotates. And I can anticipate the idea of AlphaFold by telling you that it is a prediction system which not only creates the models, but it's also aware of how well it was doing that. So we have this dark blue for model confidence very high. They have a quantity called local, I forget what it was. Well, we'll know about that in structural bioinformatics. So you can see that most of the protein core is of a very high reliability, and then we have some less reliable but still confident areas in light blue, but then these yellow and orange are of low confidence, and this is not something to wonder at. Many proteins have pieces which do not fold in an organized fashion, but they are intrinsically disordered, and so having a low quality prediction for AlphaFold models is an indication that it might be a part which is not folding at all, but it is disorganized. And then this is a secreted protein, and there is a signal peptide which is cut out, and let's see, 29, we can find where we have the signal peptide. Oh my, the signal peptide is not annotated, but it is there. So it is cut out, and then for that reason, there is no model in the databases for anything like this, and so no good prediction and no relevance to the biology if it's removed while the protein is just being synthesized. But I think this is enough of a UniProt demo for now, and I hope this gives you some hints in how to proceed the homework, and if you already did the homework, so maybe this gives you new ideas how to utilize UniProt. Any other questions you had in mind before coming here, or something that has been coming up during this discussion?
Hello, I have a question. For question
Yeah.
number, I think 18 in the homework. Wait, sorry. Yes. So it says like at which crystallographic resolution has the 3D structure of beta microseminoprotein has been characterized? How accurate is that? So I went to the PDB page
Mm-hmm.
and then for that, and how do you, because I don't work with proteins, so I don't know how to answer how accurate is that. Because for example in the PDB page I can see like a validation page of
Mm-hmm.
of the experiment. So is that sufficient to answer that question?
That tells you something about whether it's reliable, but then let's see. Let's go to PDB at RCSB, the American site. So here's one. So you can see
Mm,
that the resolution is 1.90
mm,
angstroms, and this is, in a way, it is similar to optical resolution in that the smaller the resolution, the smaller details you can observe reliably in the protein. But it is certainly different from optical resolution because you can't distinct 2 points as distinct points in a submicroscopic particle. So it is not to do with the light. We are discussing units which are smaller than the wavelength of any light. Light is at hundreds of angstroms. Angstrom is 10 to the minus 10th meters, or 0.1 nanometers. And it's been used as a unit of length in crystallography because it's close to the lengths of chemical bonds, so they become convenient numbers when you are discussing molecules. But the answer to your question is Googleable crystallography resolution. Let's see. So here we have a visual progression from really bad resolution like 5 angstrom to very high resolution 0.5 angstrom. These are kind of simulated data. So these grids are computed electron density, and you can see that when you have ultra high resolution, each sphere is pinpointing each atom really closely. Whereas when at a kind of a more realistic resolution for proteins like 1.4 angstroms, you can still see that the shapes, even if they are kind of blobs, they match the molecular shapes so that you can really confidently park your side chain of a tryptophan residue here. And you can see that the protein main chain is going here and so on. Whereas at 2.5, you probably can get the main chain, but many of these side chains are getting kind of iffy. We have some parts of the model that don't match any electron density, and we may have some kind of ghost electron density where we don't know what it belongs to. And at 3.5, this is really hopeless. You don't really have tools to create a good quality protein model out of this. And at 5 or more, you can just get the general shape of the whole protein. You can't really model it. Let's see if there is a table. Oh, yes. So here's a quick guide which tells you how accurate models are at different levels of resolution. What was the example I was looking at? That was at 1.9. So it falls in the second bracket, so that many atoms are clearly seen, but not all. So you have to kind of use your imagination and the general ideas of protein structure. But because it's proteins and we know that there are only 20 amino acids and we know the sequence of the protein, so it gets pretty easy to model, to thread the protein sequence through the observed chain of the electron density. So it's okay. But then when we are going to the level of 2.5, it says shapes of larger side chains are seen. It means that small side chains are just something you need to guess, especially on the protein surface. And what you do is you just put in the most common position for the side chain relative to the protein main chain, and so on. So you find all of these descriptions of resolution at various level, but for now it's enough to know that 1.5 is like a really good resolution, so most atoms are clearly seen, and 2.0 is actually the median resolution for proteins, new proteins which are being deposited. And so that is like fair or quite good. But then 2.5 and larger numbers mean worse resolution and less and less usability for thinking about the protein functions, because the positions of the atoms are not certain, so you have lots of room for guessing there. Okay, a long answer to a simple question. Maybe you would have been happy of just the idea that 1.5 is pretty good and 2.0 is okay. But the important thing to know is that the resolution is going to like reverse direction. So the smaller the number, the better the resolution.
The explanation was perfect. Thank you.
Yeah, you're
Thanks.
welcome. I was kind of thinking that we would be done in 1 hour. We don't have a specific time allocation. When at the time when these meetings were lectures, so that I was talking through the whole lecture topic. So it took about an hour and a half, and then we had 15 minutes for questions. But now that this is all questions and doing extra demonstrations, so we don't probably need that much time. But I'm flexible, so even if it's 8:15 now finish time, so I'm good. I walked the dog, and the dog is on the sofa and looks quite happy and tired of a long walk. So if you have any questions, so the shop is still open, but if you feel like wanting to do whatever you are doing in the evening, so I'm also good with calling it a night and stopping it for now.
I have a question that's
Go ahead,
not
yeah.
really homework related. On the website of the PASMI, at the curriculum, where the description of the subject can be found, there are 2 books mentioned as recommended reading. Are those books available somewhere?
Which one? It was the main site of the curriculum.
Yes, on the PASMI's website. So the 2 books is one of them are So one of them is bioinformatics for biologists from the Cambridge University, and the other one is Introduction to Bioinformatics. That's from the Oxford University.
Where were the names of the books? Am I in the right place even?
No, it's not here. So if you're going to the actual website of the Pazman University page.
Okay. That's I there I haven't been, but the names of the books don't ring a bell, so you should be looking at Amazon, and if they are out of print, so they might have second hand sellers as well. And if they are listed for the curriculum in the university web pages, they might be available in the university library. And again, I don't know how your library works, but in my home university, or where I'm working now, University of Turku, so there's a digital library where you can get ebooks of most of the titles. So it's been a long time since I've actually visited a physical library, but I use the digital library all the time to make use of the journal subscriptions they are paying for, and occasionally getting an ebook as well.
Okay,
But I'm
thank you.
sorry, I can't help you with the details, and I would advise you to the local people, so Zoltan and whoever might know how the book practicalities work. But for
Sure, no
most
problem.
pardon?
Sure, no problem. I just thought that maybe there's a collection of the recommended books somewhere or something like that.
No, I'm sorry, I'm not aware
Okay.
of that. And we have the course for the data analysis with R, for which Csaba Ortutai and his wife have written a book, and that is also something you can get as an ebook, and that is something that is highly recommended to the students. And again, you need to check if it's available through the library, or whether you need to find ways to buy it yourself.
Okay, thank you.
Okay, anything else?